my name's Nakia Ulrich. I work in Dr. Miller's lab. I study cyanobacteria, these cool cyanobacteria that use far-red light for photosynthesis. And so anyway, I also work in the lab that Heidi's in. She just talked about PCR and how we use that approach kind of as a copy machine to increase the numbers of DNA fragments. And so the next big step is then to sequence that DNA. And so there's many different sequencing technologies. One of them is the Sanger sequencer. This is a Sanger sequencer here. Pretty large, takes up most of the bench top. It's about over 50 years old, but still considered the gold standard of sequencing, mainly because it's the most accurate. The process with a Sanger sequencer is you have a template strand of DNA, and with it you add in dNTPs, nucleotides, and also nucleotides with the oxygen removed. And this means that when those nucleotides are added to the template strand, they stop the reaction. And they also have a fluorescent tag. And so each base has a different color. So A would have one color, T would have a different color, and so on. And so what you get is lots of different fragments of different sizes. And so when you separate out those sizes, we can look to see what the order of the A's, the T's, the C's, and the G's are based on the color of the base at the end of the fragment. So Sanger technology is one of the oldest sequencing technologies, over 50 years old, but we still use it because it's cost effective, it's very accurate, and it's great on shorter DNA fragments. And so that's why it's still used very regularly in genetics labs. It's the workhorse, you might say. So how do we look at a readout from a Sanger sequencer? Here, sequencing is currently being done. And so on this screen here is the readout of the fluorescent tag. So we're going to zoom in on that. Here's the readout from the Sanger sequencer. You'll notice lots of colors here. There's 4 colors, each for the 4 bases. And so as we go along the sequence, you'll see peaks of the different colors based on which base is at that position. So you see red and then yellow and then red. And so that might signify ACA or something like that. I'm standing next to a more recent sequencing technology, a next generation sequencer. Here is an aluminum MySeq. You'll notice that it's about the size of a cooler, so a little bit smaller than the Sanger sequencer. And these machines are really cool because you can do much larger genomes much quicker. And so you can sequence a human genome within a day, which is really cool. So another really neat thing about the aluminum MySeq is that sequencing reactions can happen at the same time. So you can have millions of reactions happening at the same time. And so the process of Illumina sequencing is you take your DNA and you break it up into fragments, and then you load it onto a flow cell. So this is a flow cell. So here's the flow cell, and millions of reactions are happening on this flow cell. And the camera is taking pictures of those reactions. And so here's a snapshot of those reactions occurring. You see lots of colors here. And so each of these are a different fragment of the template DNA, those clusters, right? And so the colors represent what base has just been added. And then the next snapshot, there might be a different color in that same location. And so millions of reactions are happening at the same time. And these snapshots of these different fluorescently tagged bases give us the sequence of those fragments. So this has been an overview of the Sanger sequencer and the Illumina sequencer. These are used very regularly by students and faculty here at UM. They live in the genome core, and they've been super important for genetics research at UM. So hope you've enjoyed.